Hi there. Okay, so this is key area 2, transport across the cell membrane or cell membrane. It's quite a long, quite a difficult one. It's worth the time investment though, because it will probably crop up in the exam. I'd say at least 7 or 8 marks. Quite often it's tied in with an experimental type question, which tests your skills like valid, reliable, accurate, those kinds of things. So it's definitely worth putting the time in to be familiar with this concept. Once you've got the hang of it, though, actually, it's really not too bad. It just requires a little bit of time and investment. So we're going to start off with the membrane structure. So we're going to have a look at the membrane that we looked at in the first key area in a bit more detail, just zooming in slightly, having a look at what it's made of.
Okay, so this is just from the SQA course specification, so it's what you're expected to know for this whole key area. I'm not going to read through it, you can pause it and have a read if you want. We're really just going to be focusing on 2A in this video, so just that first line there, and then we'll go through the rest as we go through the videos, but that shows you how big this key area is.
Okay, so things that we'll be looking at over the entire key area: cell membrane composition. So what's the membrane built of? Then we're going to be having a look at something called passive transport, and the two types of passive transport that we investigate are osmosis and diffusion. Then we'll be having a look at the effect of different solutions on plant and animal cells. And then finally we'll finish off with active transport and exactly what that is and how it works.
Okay, so again, like I've already said, in this PowerPoint, we're going to be learning about the composition, so basically what is the structure of the cell membrane. And by the end of this, you should be able to name the two main components of the cell membrane, describe how proteins are arranged in the membrane, describe how phospholipids are arranged in the membrane, and explain what is meant by the term selectively permeable.
Okay, so to remind you from the cell membrane controls what substances can enter and exit a cell. And every type of cell has a cell membrane. So animal, plant, fungal, and bacterial all have a cell membrane, and they all have this ability to decide what is coming in and out of the cell.
Okay, so in terms of the cell membrane composition, what that basically means is what is it made up of, what is its structure, but they will use the word composition in the exam, so you need to be familiar that if it's talking about cell membrane composition, it's saying what is it made up of. It's nice and easy. It's made up of two main things. It's made up of proteins and it's made up of phospholipids, which you can see labelled in this diagram here. The phospholipids, it almost looks like a ball with like a Kirby grip or something in the bottom of it. They come in pairs, so there's always one at the top and one at the bottom, with the ball as the head of it at the top and at the very, very bottom. They're pretty consistent in shape across it, you will see. And then there's proteins, which are lots of different shapes, basically these big white blobs throughout it. We're going to look at both of these in a wee bit more detail now.
Okay, these are also cell membrane diagrams. Now, cell membrane diagrams are never the same twice with the SQA. So you can see the one on the left, you've got a protein very clearly blobbing in the middle of all the phospholipids. The one on the right is a bit weirder, so the idea is it's a magnified section. Again, we're looking at the blobby bits are the proteins, whereas the things that look like tiny spoons, I suppose, that's your phospholipids. So you've got to be able to get used to the idea. It's never going to appear the same way twice. You're looking for features in common with what you know the basic shape of these should be.
Okay, so in terms of the composition, we say that the phospholipids create a bilayer, which basically means there are two layers of them, like I previously discussed. So there is always, in this case, there's a little red head with two yellow feet coming off it on the bottom, and then it's another set of two yellow feet with a little red head at the bottom of that. So it's two layers of them, one on the bottom and one on the top, and this is called a bilayer. You have to call it a bilayer, not a double layer. That's just to tell you what it means. It is a phospholipid bilayer. And then the proteins, they're a bit more random. They are kind of randomly scattered throughout. You might have an area of membrane where there's hundreds, and then you might have a similar size area of membrane where there are none. They can be totally random. They can be on the top, they can be in the bottom, they can be through the middle, they can have a gap, they can be all different shapes and sizes. So phospholipids, a structured bilayer, proteins totally randomly scattered throughout the membrane.
Some of the proteins might also notice have got like a hole through the middle, kind of like a channel or a donut. They're actually useful for what we're going to have a look at later, but the idea is not all of them can be. Some of them are just literally just blobs in the membrane. Okay, so the membrane is selectively permeable. Key term that you must know there, selectively permeable. What that means is it allows some things through and some things it does not allow through. So it's selecting what comes through. The word permeable means holey or gappy, leaky essentially, but it's choosing, so selectively, what stuff's going to allow through. Okay, so it's a very important term that crops up. If they ask you something in the exam like, what feature of the membrane allows it to control the entry and exit of substances, you say, the feature is it is selectively permeable. That's the feature that it's talking about, okay? Now, I know the exam language is horrific there, but that's the kind of questions that they can ask you, and that's the kind of response that you expect. So what feature of the cell membrane is probably going to be it's selectively permeable?
Right, so just to summarise that, this video was nice and short, there wasn't too much to it. The membrane structure, so cell membrane controls entry and exit of different materials and substances to the cell. All of the cell types have this cell membrane. The cell membrane is made up of a bilayer of phospholipids, so a double layer of phospholipids, and with proteins scattered, embedded throughout it as little blobs. And the membrane is very crucially selectively permeable, so it's picky and choosy about what it lets in and what it doesn't let in and out.
Okay, so that's what we've covered today. You should be able to name the two components of the cell membrane, proteins and phospholipids, describe how proteins are arranged in the membrane, randomly blob through it. The phospholipids are in your bilayer, so one layer on top, one layer on bottom, and selectively permeable means it can select what substances enter and exit the cell. Okay, the next video is we're going to start on passive transport, looking at diffusion.